Here are the notes for Chapter 12, Section 2, dealing with chromosomes and DNA replication. First, there are a couple things I want to make sure that you write in your notebook when you're taking these notes. The following are the vocab that are important to this section: chromatin, histone, replication, helicase, and DNA polymerase. The objectives for this section is to understand the structure of DNA in regards to its length and be able to summarize the events of DNA replication. So let's take a further look at DNA and the chromosomes. This should be all review: DNA and chromosomes. Where is DNA found? Well, reminder, the difference between our prokaryote cells and our eukaryotic cells. Prokaryotic cells, it's just going to be found in the cytoplasm because they don't have the nucleus, and they're going to have a single chromosomes, while eukaryotic cells are going to have a nucleus, and that's where the DNA is going to be found. Cells have several chromosomes depending on the type of organism. Reminder, humans have 46 chromosomes, while a housefly has 8, and a giant sequoia tree can have 22. The big key here is that eukaryotic cells have as much as 1,000 times the amount of DNA that a prokaryotic cell will have. So let's think about the amount of DNA that is actually contained in your cells. When we think about a prokaryote, E. coli has 4,639,221 different base pairs, and this all fits into one bacterial cell. While humans have almost 1,000 times that many base pairs. This means there's about a meter, around 3 feet of DNA in each nucleus of every cell in your body. Meaning the average human body, if we uncoiled all that DNA, it would stretch from the Earth to the Sun 600 times. That is a ton of DNA that is in each individual cell. So how does all of that massive amount of DNA fit into the nucleus? Well, DNA is folded around itself with the help of histone proteins. These histone proteins are what helps the DNA kind of curl up and fold. Chromatin is actually the coiled portions of the DNA and the protein histone. So if we look at the slide here, you can kind of imagine if this is our lovely DNA here. Within each of it, we have our base pairs here, our double helix. It's being folded and coiled, and that's when we get these chromosomes. So we've just talked about the massive amounts of DNA that is stored on these chromosomes. But how do we get all of this DNA, and how do we replicate it as we look at the cell division? How does this take place? Well, DNA, again, a reminder, a normal strand of DNA is in the double helix shape. Well, in order to be replicated, first it has to be unwound. And the reason it needs to be unwound has to do with this idea that each of the strands are said to be complementary of one another. And this means that the sequence on one side is the exact opposite as the other strand. And this means that even if you only see half of the chromosome, you can still create the other half. And this is how DNA replication is done. So if we look at this, if the following sequence appeared on a strand of DNA, what would be the complementary strand? What would it look like? And again, for these, you always want to focus on how are these paired. Remember, A only goes with T, and G, C, and G go together. So if you have an A on one side, it means you're going to have a T on the other side. If you have a T on one side, it means you're going to have an A on the other side. And again, this whole point, or the whole basis for replication, is based on this idea that the DNA strands are complementary of one another. So how does this actually occur then? So the unwrapping or unzipping of the DNA molecule is initiated by an enzyme called helicase. Helicase does this by breaking the hydrogen bonds that connect the nucleotide bases in the center of the DNA. So if we go back to this little picture here, and remember we have our base pairs here, and if you have a reminder, if you can kind of look, that's where our hydrogen bonds occur. So this DNA helicase actually comes in and disrupts those. And then the double-stranded structure then is able to pull apart and unwinds, exposing the nitrogenous bases of the nucleotides. So helicase splits them kind of right down the middle. After helicase unzips the double helix, another enzyme called DNA polymerase forms the complementary portion of the DNA molecule by assembling nucleotide bases onto the old DNA molecule. So what happens is, again, is you have these kind of bonds being broken, and you have these two different kind of backbones, as we will call those. And we'll use a little bit different terminology here, dealing with a strand. So replication typically is going to occur from the 5' to the 3'. What does that mean? It has to do with our backbone that we just looked at. So I'm just going to pull this open a little bit so we can see it. And what happens is you look at how the groups are arranged based on their location. So on the first strand, which we're going to call the leading strand, it goes from 5' to 3'. Well, remember, the opposite side is complementary, and it runs the opposite direction. So it'll run from 3' to 5'. And this is the lagging strand. Well, the problem is our DNA polymerase can only replicate in the 5' to 3' direction. So that causes a little problems when we look at how do we get both strands replicated properly. So the lagging strand can only be completed in smaller pieces. And what happens is that we have these small chunks on the lagging strand known as Ozaki fragments. And they're basically there because this replication can't occur between the 5' and the 3'. All right, now that I've probably thoroughly confused many of you, let's take another quick look at this idea of replication. So replication, its goal is to take this DNA and replicate it. So we have our helicase that's going to come in and split it, and we're going to get what's called this replication kind of fork occurring, where we're separating the two strands by breaking those hydrogen bonds. And that's all fine and dandy for our leading strand. And think of the leading strand as the one that has it easy. It goes in the 5' to 3' direction. So the DNA polymerase has no problem with that. Well, unfortunately, the lagging strand runs in the opposite direction. So what actually kind of occurs is the replication is done in segments. And you have this RNA primer that's basically going to jumpstart the entire process. It jumpstarts the process for the leading strand, but it's going to have to do it multiple times for our lagging strand. And so what happens is this primase comes in at multiple sections to give us the right kind of direction, and then it'll work backwards. So it basically gives it a starting point for the DNA polymerase to go ahead and continue replication. So that's why we get all of these different kind of fragments depending on where the RNA primer gets it started. So at the very end of everything, now that the RNA primer has started, the DNA polymerase can go ahead and finish completing that lagging strand. But that's where all those little pieces come, and you can just think the lagging strand is just a lot harder for replication. Again, the new strands of DNA are constructed with the DNA polymerase, and the two old strands serve as the template for the new strands to be constructed. And each new DNA strand is made up of one new strand and one old strand. Each of the new strands must be an exact copy or else a mutation will occur. And again, mutations only occur in about 1 in every 10 billion of these replications, but mutations can cause serious problems for not only the cell, but possibly the organism the cell is in. So again, the body has kind of checks and balances with this idea of errors. And this is where the DNA polymerase comes back into play and kind of proofreads the entire process to make sure that there were not any errors, making corrections. So this ends the video notes for chapter 12, section 2. From this, you should be able to explain how DNA is replicated, how are the long DNA molecules found in eukaryotic cells, packed into short chromosomes, what is the role of the DNA polymerase in DNA replication? And how is the structure of chromosomes in eukaryotes different than the structure of chromosomes in prokaryotes? So you should be able to go back and answer these. You should also fill in all of the vocabulary information and make sure you understand those objectives. Here ends the notes.